Today I am going to give you an idea about the types of yeasts and their basic characteristic features. The term yeast has no taxonomic standing of its own and simply denotes a growth form in several groups of unrelated fungi. The English word yeast means foam and to rise, which directly relates with fermentation process and fermentative Ascomycetes fungi, Saccharomyces cerevisiae. The discovery of some taxa that are Basidiomycetes has broadened our perception of the nature of yeast. As a consequence, we have come to know yeast as fungi, the vegetative state of which reproduce by budding and fission and results into single cells. All of you know that there are some fungi which are dimorphic in nature, that is, shift from their hyphal state to yeast-like growth under certain environmental conditions. Just to demarcate those dimorphic fungi from the yeast, I think it is give a definition of yeast in a proper manner. So, yeast now may be considered as a fungi which predominantly multiply by budding and fission and which do not produce their sexual state within or upon a fruiting body. Traditionally, yeasts are classified and identified by their cellular morphology and also by a set of standardized fermentative and assimilation tests. However, the phylogeny of yeast is uniquely illustrated by their assignment in two different classes, that is Ascomycetes and Basidiomycetes. Subdivision of taxa within their respective classes is made by the comparisons of morphological as well as physiological features. Nowadays, application of molecular comparisons offers an unprecedented way to re-evaluate the existing system of yeast classification. As in contemporary fungal classifications, the anamorphic, asexual or imperfect yeasts are classified into Deuteromycetes till such time that their perfect state is discovered. However, affinity of such yeasts are determined by a number of ways. Say for example, Ascomycetes yeast do not contain xylose in their cell wall and their cell also lack pyrophosphatidic acid, while Basidiomycetes yeasts contain xylose in their cell wall and pyrophosphatidic acid in their cell. Budding in Ascomycetes yeast is basically bipolar or multilateral in nature, while in Basidiomycetes yeast it is entirely enteroblastic. Anyhow, other characteristics like presence of blastoconidia and red carotenoid pigment in the body also indicate the affinity of yeast towards Basidiomycetes. Today I am going to give you an outline yeast classification adopted from the book The Yeast, a Taxonomic Study by Glucomannan and Fell, published in the year 1998. considering the phylogenetic diversity of yeast, they may be divided into two main group, that is teleomorphic and anamorphic Ascomycetes yeast and teleomorphic and anamorphic Basidiomycetes yeast. Ascomycetes yeasts are usually classified into three classes: Archaeascomycetes, Euascomycetes, and Hemiascomycetes. The class Archaeascomycetes divided into four orders: Schizosaccharomycetes, Taphrinalis, Protomycetes, and Pneumocystidaceae. While the Euascomycetes group contains only one genus, that is the Oisporidium. However, the Hemiascomycetes class include only one order, that is Saccharomycetes, which includes 11 different families like Ascoideaceae, Cephaloascaceae, Dipodascaceae, Endomycetaceae, Eremothicaceae, Lipomycetaceae, Metzincoaceae, Saccharomycetaceae, Saccharomycodaceae, Saccharomycopsidaceae, and Candidaceae. Whatever it may be the classes or order included under the head Ascomycetes, usually their basic characters may be of different types. For example, septal morphology has been considered as a preliminary phylogenetic characteristics for differentiation between Ascomycetes and Basidiomycetes yeast. In Ascomycetes, the septa is of simple porous type and the septal wall is always electron transparent and homogeneous. Through the large central pore, usually the nucleus can pass from one cell to another. But there are some members where only the micropore septa are present, where the pores may remain plugged or may remain unplugged. In the picture you can see that a nucleus is migrating from one cell to another and the septal wall is clearly electron transparent and homogeneous. The next important characteristic feature of Ascomycetes yeast is the response of yeast colony towards the reagent diazonium blue. In this reagent, the colony takes no color, that is, Ascomycetes yeast remain unstained when treated with diazonium blue B. The next characteristic is the urease reaction. Ascomycetes yeast usually fail to hydrolyze urea and thus give negative reaction towards hydrolyzation test. The next important characteristic is the cell wall structure. And the cell wall structure is usually indicated by the electron microscopic pictures, particularly at the budding site. In Ascomycetes yeast, the inner wall of the cell is typically uniform and homogeneous, as you can see in the photographs. At the budding site, usually two closely appressed walls are visualized, of which one is parental and another one is the emerging bud's cell. The next vital characteristic is the polysaccharide composition of the cell wall. In the diagrams, you can see that how the polysaccharide components remain arranged within the cell wall of Ascomycetes yeast. The major component of Ascomycetes yeast cell wall is the alkali insoluble, acid insoluble beta 1,3 glucan. Along with this beta 1,3 glucan, very low amount of alkali insoluble, acid insoluble beta 1,6 glucan remain present. However, very small amount of alkali soluble beta 1,3 glucan remain impregnated. The next major wall component is glucomannan protein complex, and a small chain of beta 1,6 linked glucose residue enter into the protein moiety of mannoprotein for anchorage and cross-linkage between the glucan chains. Chitin remain present in very low amount, that is around 1% of the total, and they remain restricted on the bud scar area, particularly you can see in the next diagram. Other important characteristics like cellular growth also useful for distinguishing Ascomycetes yeast from Basidiomycetes 1. Yeasts are basically unicellular, typically uninucleate members. Actually, this unicellular plant body produce new cells serially, as you can see in the budding yeast or Saccharomyces cerevisiae in the picture. The next important characteristic is the formation of bud. In Ascomycetes yeast, the budding usually takes place from a particular locus, and the second bud never arises from the same area. That means one bud per locus mode of budding limits the number of buds produced by a parent cell. Actually, bipolar budding is observed in some members like Saccharomyces, where the next bud is produced just from the opposite end of the cell and not from the same area, as you can see in the diagram. Another type of budding is observed in Ascomycetes yeast is multilateral budding observed in Protomyces, Lipomyces, Zygozyma, Candida like members. However, in this diagram you can see a color transmission electron micrograph of a budding yeast. The process of budding in yeast always starts after softening and thinning of parent cell wall. After enzymic thinning and softening of parent cell wall, the protoplast remain covered by a thin, delicate structure through which it formed a small outgrowth which finally matured into a bud-like structure and converted into a full-grown bud in course of time. After formation, this bud separates from the parent cell and leave scar on the parent cell as well as on the bud itself. In this case, you can see that the bud scar is always concave while the bud scar is always convex in nature. This is also a color diagram of budding yeast cell, a transmission electron microscopic view. Nuclear division in Ascomycetes yeast takes place within the parent cell. After nuclear division, whenever the daughter bud is formed, it transferred into the bud proper. In the diagram, you can see that the separation or splitting of the spindle pole body takes place within the parent cell, and nuclear elongation takes place within the parent cell, and whenever the new daughter nucleus is produced, it is transferred to the bud proper. This is something different from the Basidiomycetes yeast. The next characteristic feature is the guanine and cytosine content of the nucleus. In Ascomycetes yeast, the G plus C content usually ranges between 27 to 50 mole% in contrast to Basidiomycetes content above 50%. The next important characteristic is the sexual cycle. Ascospore formation starts always after meiosis, and during meiosis, what happens? The spindle pole body initially divides and migrate into the opposite pole, as you can see in the diagram. But after initial elongation of meiosis 1 spindle pole body, the spindle pole bodies divide further and arrange themselves within the nuclear envelope at the four corners in a precise, orderly manner. After arrangement of the spindle pole bodies, usually a small cup-shaped structure encapsulate the nuclear material and then entirely separate the four nuclei formed thereafter. In the next stage, the wall materials are deposited in between the two membranes, and finally ascospores wall are differentiated. The primary ascospore wall is basically electron transparent, uniformly thick, but secondary ascospore wall materials are typically patterned and deposited outside the primary wall. All of you know that strains of Saccharomyces cerevisiae produce a toxin called killer toxin, or technically the mycosins, which are proteinaceous or glycoproteinaceous nature, and they are capable of killing the strains of similar or closely related species. Actually, this determinant is cytoplasmically inherited, that is, this toxin producing ability transmitted from one generation to other via cytoplasm, that is, it indicates the maternal inheritance clearly. This killer system is usually regulated by the double-stranded RNA or linear double-stranded DNA plasmid or by chromosomal genes. Considering the proteinaceous nature or glycoproteinaceous nature and the involvement of cell wall components of fungi, it is generally deduced that this killer system or the mycosins may be used as a taxonomic tool in near future. However, the mycosin producing strains are resistant towards their own toxin, but they can easily kill the strains or species of closely related species, as I said earlier.